I remember a season when the world slowed its breath. Streets that usually hummed with commerce went silent. Airports, markets, classrooms, all the ordinary theaters of life grew still, as if the planet itself had taken a moment to listen. In that pause came a challenge the modern world had never met before. A new virus moving through human populations with a speed and consequence that forced the urgency of governments, hospitals, and laboratories into a single, shared tempo. Fear and grief were immediate, so was resolve. What followed was not a miracle in the sense of the inexplicable, but something else. The rapid, deliberate unfolding of a vast global experiment in cooperation. It was the race for a vaccine. The first step of that race was simple and small, and it happened at the level of code. Within days of clinicians identifying an unfamiliar pneumonia, scientists read the pathogen's genetic instruction manual and placed it on the public table. That act, sharing sequence data in the open, removed the first lock. Across continents, researchers could now see, in letters and patterns, the shape of the enemy. Sequencers in quiet labs and high-throughput facilities hummed. Bioinformatics pipelines that had once taken weeks ran in hours. The virus became known not just by its symptoms, but by its precise molecular architecture. But knowing a genome is not the same as stopping a disease. For that, decades of quiet, cumulative science had to be summoned. Work on coronaviruses after earlier outbreaks, research into immune signaling, and a long arc of innovation in vaccine platforms, some of them obscure and underfunded, were suddenly thrust into the spotlight. mRNA technologies, which had been coaxed forward by patient experiments and incremental breakthroughs, offered a new route, a way to tell the body's own cells to make a tiny, safe fragment of the virus, and in doing so, teach the immune system to recognize and fight it. Other approaches, viral vectors that delivered genetic blueprints using weakened viruses, inactivated whole virus vaccines, protein subunits presented with novel adjuvants, ran in parallel. This diversity mattered. It was not a single idea that would win the race, but a chorus of strategies moving in concert. What made the pace extraordinary was a set of practical choices that rewired the usual arcs of vaccine development. Traditionally, vaccine research followed a linear march: design, small-scale safety testing, larger efficacy trials, regulatory review, and then, if all went well, manufacturing and distribution. In this moment, the steps did not vanish, but they overlapped. Clinical trials that would usually wait for long-term safety signals were designed to run at scale, while manufacturing capacity was built at risk. Regulators created new pathways for emergency use, demanding rigorous safety and effectiveness data, even as they acknowledged the emergency. Funding that often takes years to assemble appeared in months, driven by governments, philanthropic organizations, and consortia created precisely for this purpose. When the world was on the line, the normal calculus of risk shifted. Companies and governments elected to produce millions of doses before final approval, accepting potential losses to shave weeks or months off delivery timelines. Behind these technical decisions were 1,000 unspectacular scenes. Researchers pipetting by lamplight, clinicians recruiting volunteers at local clinics, statisticians wrestling with trial designs that could provide clear answers fast, community health workers explaining participation to neighbors, supply chain managers calculating how to move vials that needed temperatures colder than a winter night. Preprint servers and open databases became the agora of science. Papers that might once have been sealed in paywalled journals were posted and debated within days. Data moved at the speed of the internet. The Global Genetic Surveillance Network, labs tracking viral mutations and sharing sequences, kept pace as variants emerged, informing whether a vaccine's target might be shifting underfoot. The human scale of the effort was enormous. Tens of thousands of volunteers enrolled in trials, many motivated by the simple wish to help. Hospitals became not only places of acute care, but hubs of research. Manufacturing floors that had once made other products were retooled. New facilities were stood up. Glass vials and stoppers, lipid nanoparticles, and cold-chain freezers became as vital as microscopes and centrifuges. Logistics teams learned to dance with perishable biology. Trucks outfitted with dry ice, planes chartered to carry temperature-controlled cargo, clinics set up in gymnasiums and community centers. And everywhere, clinicians and scientists worked under stress and scrutiny, balancing speed with a duty to safety. There were setbacks and controversies, supply bottlenecks, geopolitical tensions over access, debates over prioritization, and the human drama of rare but real adverse events that had to be investigated and understood. Misinformation threaded its way through social media, sometimes slowing uptake and complicating public health messaging. Yet the technical achievement remained undeniable. From the first shared genome to real-world immunizations, the timeline that used to take years compressed into months. The first authorizations for highly effective vaccines arrived in under a year, a rate of progress unthinkable in earlier eras. Those months were not free of mistakes, but the institutions of science, peer review, regulatory oversight, surveillance systems, functioned under pressure to hold the line. A key reason this success was possible was the platform nature of some vaccine technologies. An mRNA sequence could be designed on a computer, synthesized quickly, and packaged into the same delivery system that had been refined for other applications. That meant once the viral target was known, teams could move from design to trial-ready product in a matter of weeks. Meanwhile, other candidates used well-understood manufacturing processes that could be scaled by companies with decades of experience. Multiple winners were not just likely, they were essential. Different vaccines, different logistical realities. Some required ultra-cold storage and were ideal for urban hospitals. Others could be shipped in standard refrigerated trucks and used in rural clinics. The plurality of approaches increased the global system's resilience. The race did not end with approval. Distribution became a new test of coordination and equity. Rich nations secured early supplies, while coalitions and funds were created to channel doses to lower-income countries. Programs designed to ensure fair access grappled with real-world shortages and political friction. Yet even here, partnerships across governments, industry, nonprofits, and local communities produced creative solutions. Shared procurement mechanisms, technology transfer initiatives, and the building of regional manufacturing capacity that began to reshape the landscape of global health production. As vaccines rolled into arms, the world saw another kind of data, the quiet arithmetic of lives saved, hospitals spared, and economies nudged toward recovery. Real-world effectiveness studies, surveillance of variants, and booster strategies became the next chapter. Science, never static, adapted once more, learning from each viral surge and each immunized population. New variants challenged assumptions, prompting updates in surveillance and vaccine design. The speed of initial development set a new baseline. The iterative cycle of monitoring, adapting, and redeploying became the norm. If this story is, at its heart, about techniques and timelines, it is also about a less tangible reality: trust. The fastest collaboration in history succeeded because millions of strangers, the technicians, the regulators, the drivers, the volunteers, accepted a blend of urgency and uncertainty and acted. They accepted an imperfect world and chose to make it better anyway. They built bridges between public and private, between academia and industry, between countries with different languages and laws. The crisis did not erase geopolitics or inequality, but it did reveal how much could be achieved when knowledge was treated as a common good. Looking back, the race for the vaccine reframes a single, stubborn truth: scientific progress is cumulative. No breakthrough appeared ex nihilo. The rapidity was possible because of investment in basic science, long-term funding for platform technologies, international data sharing, and systems built after earlier epidemics. It was also a lesson in humility. Speed required transparency, continuous monitoring, and the willingness to pause, investigate, and adjust when signals demanded it. There are practical lessons to carry forward. Sustained investment in global surveillance and in manufacturing capacity in more parts of the world can shorten response times in future crises. Open data practices and clear communication can build public trust before the next emergency. And the social contract between science and society, how communities partner with researchers, how regulators balance urgency and caution, how distribution is governed, remains as important as any technical advance. The story of this race is not merely a record of vaccines produced or doses delivered. It is a portrait of what becomes possible when urgency meets preparation and when competition is tempered by cooperation. It shows the architecture of modern science, networks of expertise, tools built over decades, and institutions that can flex under pressure. It shows the human capacity to move at scale, not always perfectly, but with purpose. In the months and years after the first doses were given, the world did not return to a previous normal overnight. There were new debates, new disparities, and new questions about how to live with endemic threats. Yet the achievement stands. A global crisis produced a response that rewrote the timelines of possibility, proving that when knowledge is shared and resources are aligned, what once seemed impossible can be made routine. Time remembers the long nights and the long calculations, the freighted silence of battered hospitals, and the small triumphant sound of a vial being opened. And time reminds us, perhaps most profoundly, that preparedness is neither spontaneous nor free. It is the quiet labor of many years, multiplied by the conscience to act together. The lesson hangs in the air like an echo that refuses to fade. When the world decides to move in the same direction, science can run faster than fear.